Hello everyone. Today we are going to talk about Introduction to Next Generation Sequencing. Let us begin with the discussion points first. Now, today we will focus on points like what is DNA sequencing, then evolution of sequencing, which will cover the history of DNA sequencing, the era of sequencing. Then we will move to what is meant by next generation sequencing, why is it important, different platform and technologies and applications. We will also be discussing on some aspects of NGS data analysis, which will include points like the general pipeline and different workflows, um, sample processing, file types and tools used. And lastly, we will be summarizing the session. We will discuss the future perspective. Okay, so now let's move to next slide, that is DNA sequencing. Now, DNA sequencing is to figure out the order of DNA nucleotides or the bases, which we call as adenine, thymine, guanine, cytosine in a genome that makes up an organism's DNA. We all know Dr. Frederick Sanger. He is the father of DNA sequencing and won 2 Nobel Prizes for his work on DNA and protein sequencing. Now, we all know that he is well known for determining the amino acid sequence of insulin by Sanger sequencing at the Sanger Center. Now, Professor Sanger first tramp was to determine the complete amino acid sequence of the 2 polypeptide chain of bovine insulin, the chain A and chain B, in 1952 and in 1951 respectively. And then he turned his work towards DNA sequencing. Now in 1977, Sanger and his colleague introduced a dideoxy chain termination method for sequencing the molecules, also known as Sanger method. You can see in this picture the 3 basic steps of automated Sanger sequencing. The figure as it shows, the first step is the DNA sequence for the chain termination by PCR. The second step is the size separation by gel electrophoresis. And the third step is the gel analysis and determination of the DNA sequence. Now, the sequencing method is based on the selective incorporation of chain terminating dideoxynucleotides by DNA polymerase during the in vitro DNA replication. It then forms extension products of various lengths terminated with dideoxynucleotides at the 3' end. The extension products are then separated by capillary electrophoresis. Okay. Now, Alan Maxam and Walter Gilbert published a DNA sequencing method in 1971 based on the chemical modification of DNA and the subsequent cleavage at specific bases that requires radioactive labeling at the 5' end of the DNA and the purification of the DNA fragment to be sequenced. Later on, the chain termination method developed by Frederick Sanger became the method of choice owing to its relative ease and reliability, and they used fewer toxic chemicals and lower amounts of radioactivity than the Maxam and Gilbert method. Eventually, the chain termination method was soon automated and was a method used in the first generation of DNA sequencers. Okay, so now let's move to the next slide where it will take you to understand how the evolution of sequencing has taken place. Right from evolution since 1953, when Watson and Crick discovered the structure of DNA double helix, till the era of the next generation sequencing, that is the 454 sequencing or Illumina sequencing, etc., various researchers began to adapt different methods in order to sequence DNA. Okay. Now, the development of recombinant DNA technologies, or if you can say the molecular techniques such as the PCR, which was discovered in 1985, it further helped and aided in the genomic revolution to providing means of generating the higher concentration of pure DNA species. As you can see in the timeline, in 2008, heliscope sequencer was designed and developed, and it was developed by Stephen Quake Research Group, which is also a loop chip sequencing equipment. In the same year, nanopore sequencing also came and it was developed based on the electrophoresis technology. In the next year, single molecule real time was developed. In 2010, ion PGM system for next generation sequencing were put into use. The ion semiconductor sequencing makes high quality next generation sequencing and is available and accessible to all now. Now, this evolution has taken the science into a revolutionary state. Okay, over the years, innovation in sequencing protocols, the molecular biology, and the automation increased the technological capabilities of sequencing, while decreasing the cost, the toxins, the chemicals, allowing the reading of DNA hundreds of base pairs in length, the massively parallelized to produce the gigabases of data in one simple run. Okay, so again, moving to the timeline, we can see here the slide has presented the DNA sequencing timeline. Okay, the evolution has got the researchers moved from the lab to computer-based analysis, from poring over gels to running codes. Okay, so some of the most revolutionary and remarkable events in DNA sequencing is the next generations, the PCRs, the SMS, which we call the single molecule sequencing, then sequencing the lower limit. These new approaches targeted to complement and eventually replace Sanger sequencing. This was taken from the reference Ruth Pereira et al. In 2020. It's a very recent paper from the Journal of Clinical Medicine. As you can see here in the reference here, the reference link is given below, students. So yeah, it's a very good reference and I would request everyone to go through this timeline. Now coming to the next slide, we will just understand the first, second and the third generation of sequencing. Now let us begin the first generation of sequencing, which is referred to either the Sanger, the chain termination method, or the Maxam-Gilbert sequencing method. These methods include radio or fluorescently labeled ddNTPs nucleotides of a given type, which once incorporated prevent further extensions, and these are included in the DNA polymerization reaction at low concentration. Okay. All in all, the first generation of sequencing technology has the read length ability of less than 1000 base pair. Eventually, newer dideoxy sequencers such as the ABI Prism range developed and produced by the Applied Biosystems. However, its high cost and low throughput and other disadvantages result in serious impact of its real large scale application. If we go to the second generation sequencing, we can see the DNA sequencing parallelized amplification. What happened is that in the DNA molecule being clonally amplified in an emulsion PCR. Okay? Adapter ligation and PCR product DNA libraries with appropriate 5' and 3' end, which can then be made single-stranded and immobilized onto the individual suitably oligonucleotide TED microbeads. Now, this DNA polymerase can then be added to the wells, and each nucleotide can be washed over in turn. And then the dNTPs incorporation monitored and then later visualized by detecting the fluorescent reversible terminator nucleotides at the end of the proceeding extension reaction and requiring cycle by cycle measurements and removal of terminators. Now, this is the conceptual basis underlying the sequencing in 454 ion torrent breach amplifications to produce cluster of clonal DNA population in a planar solid phase PCR reaction, as occurs in the Solexa or Illumina sequencing. In early years of second generation sequencing, perhaps the third major option, along with the 454 and Solexa Illumina, was sequencing by oligonucleotide ligation and detection from the applied system, which became life technologies following with a merger with Invitrogen. I'm just giving you an idea about the companies here. Now, coming to the third generation sequencing, we have single molecule real-time SMRT. This, which is a parallelized single molecule DNA sequencing method. A single DNA polymerase enzyme is affixed at the bottom of ZMW, which we call as zero mode waveguide, with a single molecule of DNA as a template. Now, this is a feature we can find in PEG biosequencers. Next generation sequencing, which is also known as high throughput sequencing, include Roche 454 sequencing, virtual terminator sequencing, solid sequencing, Helicos Biosciences, which work broadly in the same manner that Illumina does. Okay. And if we talk about pyrosequencing, which is a non-electrophoretic and bioluminescence method, it measures the release of inorganic pyrophosphate by proportionally converting into visible light using a series of enzymatic reactions. Okay, it generates large read length, unlike the conventional ones. Solid again uses the ligase-based sequencing. Instead of DDNTPs, oligonucleotides and probes are used instead of DNA polymerase. The library is an adapter ligated fragment library that can be used as an emulsion PCR approach, but it has a short read length. Now, these are the recent technologies, okay, allow us to sequence DNA and RNA much more quickly and cheaply than the previously used Sanger sequencing, and thus have revolutionized the study of genomics and molecular biology. Okay, the features for this next generation sequencing, which include high throughput DNA sequencing technique, or low sample size, or low reagents, or produce enormous amount of data, or high specificity and sensitivity, are mentioned over here. And the advantages of NGS technologies included are the features of this. Okay, now coming to the next slide, I will show you that there are different platforms of sequencing that can be performed. See the picture over here, it shows you the different platforms. That means the different sequencing platforms. As you can see here, the slide shows the NGS platforms of most trusted names. The NGS today are Illumina MySeq, NextSeq 500, HiSeq 2500, HiSeq 4000, HiSeq X sequencing technology. Then we have Life Technologies, IM Torrent sequencing technology, Pacific Biosciences, which in shortly we call as PEG Bio. And sequencing methods vary among them as it differs in template preparation, sequencing, and imaging, and also in data analysis. Now, the difference between all these NGS platforms are based on read length, read per run, accuracy, and runtime. And there are also cost effectiveness also play a role for each different platforms. And each platforms have their own advantages and disadvantages, okay? And so different researchers made their choice accordingly, like which platform to be used. Now coming to the features of the NGS technologies, let us understand how this evolved NGS technologies are better than the conventional classical sequencing. Now, NGS technologies have progressive advantage in terms of cost effectiveness, unprecedented sequencing speed, high resolution, and accuracy in genomic analysis. From the table, it is clearly understandable that based on the features, that is how samples are sequenced, data collection, effectiveness, library preparation, NGS has got many advantages, and thus they are preferred. We can see in the NGS high degree of parallelism in terms of sequencing compared to the low throughput in the capillary method. Reagent cost is there, which has come down comparatively, and also the sample size, which is very low in NGS. One can load very small sample to obtain the massive amount of data in a very less time. Let us also see what different applications of NGS technologies are. First being the DNA sequencing. gene regulation analysis, sequencing-based transcriptome analysis, detecting SNP or variations discoveries, as NGS capture a broad spectrum of mutations than Sanger sequencing. It is a very popular for mutation detection. Then in cytogenetic analysis, chip sequencing, and small RNA discovery analysis. Now, the figure shows the versatile applications of NGS technology in pharmaceutics in the left, and on your right, you can see the timeline of major achievements in sequencing technologies in medical and healthcare, especially in personalized medicine. For example, if we talk about identifying disease-associated mutations enabling accurate diagnosis for patients' disease, NGS has provided a powerful platform. Okay, it provides comprehensive genome sequence information. Similarly, in the case of antibiotics or chemotherapeutics, this enables drug response and the mechanism of drug resistance to be determined, so that the correct therapeutic intervention can be determined. In pharmaceutical sciences also, in medical field, NGS has helped in the identification of newest target and understanding the signaling pathway associated with disease or any kind of conditions. NGS concept used for transcriptome sequencing gives us gene expression profiling of target population and can lead to the identification and characterization of differences in the transcript abundance at genetic level. This can be correlated to have an overview of the transcribed product in response to vaccination. Thus, NGS technology has offered a way to develop safer vaccines, and in the future, it's very likely to take the vaccinology into the era of pre-vaccination genome-wide screening to generate personalized vaccination regimen. Okay. Now, again, we have come so far and have understood the NGS concept, its history, the technologies, various platforms and application. Before going to understand and take up your training on NGS analysis, let us learn a few concepts for the same. Some important points like workflows, pipelines, file types, tools employed, we need to understand, and this is very important. Okay, so let's begin. Now, NGS technologies such as WGS, RNA-seq, WES, ChIP-seq, or metagenomic generate a significant amount of data output. And here, let's take a moment to go through the basics of sequencing analysis. There are different workflows in the NGS, and each workflow has its own pipeline depending on the aim and objectives of the research study. To help you understand the process involved in NGS, let us take an example of RNA-seq differential expression profiling pipeline. Okay. Basically, what happens is that the wet lab scientists perform the nucleic acid extraction, fragmentation, reverse transcription, library construction, and then the samples are loaded in the sequencers or in the platform, the sequencing machines for the sequencing. Okay. Now we call this as experimental biology. Now, being the bioinformatics analyst or scientist, the computational parts come into play. Okay, to perform the post-sequencing analysis, such as starting from the sample processing to check their quality. Now, depending on the aim of the study, one would choose the RNA-seq analysis, where DEJ analysis pipeline will be followed. Further, it moves to the system biology for more insights and research. Okay, I hope this is clear now, how the workflow actually works. Now, most importantly, I would like to bring your attention that each and every NGS application like variant analysis, metagenomic, ChIP-seq, exome sequencing, DNA methylation analysis, or microRNA analysis, etc., have its own pipelines. Okay. Now, what we have seen, this is the workflow, and each workflow has their own pipelines. By the pipeline, actually, it means that each NGS workflow has its own series of processes, usually linear, to transform data. Okay. Example: the first process takes raw data as input, We do quality check and then send its result to the second process and so on, and eventually ending with the final result in the pipeline. One needs to learn and gain a good knowledge on the tools and execution of each and every pipeline. Okay, I hope this workflow of the example taken is clear now, how work from experimental biology to computational biology to system biology and further biological insights are performed. Okay, now let's move to the another example of the NGS workflow. As we can see in this picture, excuse me, genetic variant analysis. We can see here a sample is taken from, let's say, a human gut sample or any microbial species, which is needed to be sequenced. Okay, the first thing we need to do is the sequencing data. The sequencing data is to assess the quality, the whatever data it is generated from the sequencing, it's need to be checked for the quality. That means we need to assess the quality of raw sequencing data. Then some pre-processing procedures has to be done to improve the initial quality of the data. The next step is mapping and also called as aligning. Okay. Aligning of your reads to the reference genome or the reference transcriptome. It is a good idea to check again the mapping quality, as some of the biases in the data would show after the mapping step. Okay. Now then variant analysis performed, including variant calling or the prediction or the predicting the effects of the identified variants that is produced from the known genes. Example, amino acid change or frame shifts or deletions, whatever. One can go for, again, visualizing the data. The standard tool for visualization of map reads and the identified variants is the genome browser. So this is another example I have showed you to make you understand how the pipeline works for if we take a genetic variant calling method. Now, again, in this slide, we will see here the different files generated from the different steps in NGS pipeline and how the file actually looks like. Okay, so there are a lot of file formats related to NGS analysis. The different sequence platform, the different sequence related formats include different information about the sequence. Okay, so NGS file formats of raw data usually come in FASTQ formats. So what we see in NCBI SRA database are the gene bank formats, that is the SRA formats. The alignment produces the SAM file, which we call a sequence alignment map, or BAM file, that is binary alignment map file. Genome browser formats are always in BED format, which is browser extensible data format, or GFF format, that is gene transfer file format, that are annotation files. Then variant analysis produces VCF file, as you can see here from this workflow, which we called it as variant call format files that contains the SNP or index. Then we have SRA file formats in which all NCBI SRA content is provided. Then we have SFF formats. The SFF stands for standard flowgram format files, which that are from the 454 equivalent to the ABI chromatogram files. Okay. Now, as we can see in the picture, the different processes like pre-processing of reads for amplicon analysis, the raw file generated from the sequencing platform are subjected to demultiplexing or adaptive trimming or quality filtration. Okay. Now, example, if I have to take like in demultiplexing, the reads are assigned to their sample of origin based on the sequence of their corresponding molecular barcode. Now, these barcode reads and the cell-free DNA reads are written, or you can say it is converted to different FASTQ files. And there are various tools for that, okay? Then different sequencing platforms will produce different file formats, and there are tools again to convert them for their respective analysis. Now, we can also see here that different platforms actually produces different different files. Like for example, we can see here Illumina actually it gives different files like FastQ, QSEC, then Roche 454 also give BAM, SFF, FastQ. So different machine specific informations are provided in this box, and the reference to it is also provided here, where it is to bring to your attention that all the sequencing platforms actually produces different file formats, and then demultiplexing is done in primary analysis step, where we need to convert our files to the FastQ. Okay, because the most common file format in the NGS world is the FastQ. Now let me show you an example. The FastQ format was developed to provide a convenient way of storing the sequence and the quality score in the same file. Now Illumina sequencing always gives you a FastQ file. These files are almost identical to the Sanger FastQ files, but the encoding for the quality score is a little bit different. There are now, if you can see here, I can show you in the picture here, it's a FastQ file format, where you can see there are 4 lines for one read in FastQ file. Okay. The first line being the sequence identifier. The second line is the sequence, followed by a spacer or a separator. As you can see here, the plus sign in the third line. And then we have some ASCII codes assigned in the fourth line, which internally depicts the Phred scores or the quality scores. When we start working on our pipelines, you will understand more clearly how the file handling works in NGS. Okay. So this is an overview about different machine sequencer machines that produces different type of file formats. The different pipelines will generate different file formats, and there are tools, of course, which will help you to convert one file to another file format. And obviously, I have shown you the example of how a FastQ file looks like. Okay, and then summarizing our NGS introduction today, we can say that NGS is an extremely powerful tool for conducting basic biological research, and it offers unmatched opportunities for improving diagnostics, prognostics, and targeted therapies. Now, the application of NGS is in translation of research findings from bedside to clinic is ever growing, is very much rapidly increasing. This is necessitating the development of computational tools to manage the growing wealth of sequencing data that it produces, and again, in turn, the efficient analysis and interpretation of enormous databases of information drives and directs the development of safer drugs with greater efficacy and less toxicity to improve patients' health and preventing disease, of course. And we will be very happy to give training in the field of NGS data analysis, and for any queries or meeting, please contact us at the address mentioned here. We have a team of experts who give training and provides an array of services. The email IDs are also mentioned here. I hope this was very helpful, and thank you for your listening. Thank you so much.